Now, for chapter 3, we're going to take a brief look at bacteria and archaea. Prokaryotic cells, prokaryotic meaning do not have a nucleus. No nucleus, no internal membranes, no organelles. When we look at these bacterial cells, we're going to do so by grouping them into 3 categories. External, these will be appendages like cilia, fimbria, flagella. Any type of specialized surface layers they may have, a capsule or slime layer. Then we're going to look at the boundary. This is going to be the cell wall, that peptidoglycan layer primarily. May touch on the mycolic acid. How thick is it? Is it thick for a gram-positive, thin for a gram-negative? Going to talk about the cytoplasmic or an outer membrane, depending upon how many membranes I have wrapped around the cell. And then going to touch on some of the internal stuff. There's not much there. Some DNA, some ribosomes. But we'll talk about that at the end of the chapter. So when you look at this figure from the textbook, it's a little hard to see here on this slide, but they've listed out all the components. When you look at it and you compare it to some of the figures and pictures you've seen of eukaryotic cells from previous biology courses, either at Lindenwood or back in high school, there's really not much going on here. I mean, look at it. No internal organelles. There may be a couple of crystals or things like that where proteins have been glommed together. But notice, no nucleus. The DNA here is just kind of bundled up. The spots you see everywhere are ribosomes. The big thing is, how many layers does it have in its boundary? Cytoplasmic membrane, maybe a cell wall, an outer membrane, a capsule, and maybe some things sticking out. And we'll talk about each of these. First, though, we got to get some nomenclature. Remember from a previous lecture, I told you nomenclature. You have to use the right words at the right time. So we're going to talk about some of the correct words to use. Now, as we're talking about these, it's going to be you're going to be dealing also with arrangements, not only the shape of the cells, but arrangements. Unfortunately, a lot of times students think that just because the bacteria may arrange together, all bacteria function as independent organisms. That means they may act in concert to a bit, formation of biofilms and things like that. But when it comes right down to it, it's every cell for itself. They may clump together as a means of protecting the species, protecting what's there. But it comes right down to it, they're not sharing nutrients. Not like your cells in a tissue and an organ will pass things back and forth. Bacteria cells do not do that. So, nomenclature. These are words that you need to know, and these are words that from here on out you will have to use. Instead of saying a bacteria is shaped like a sphere, you're going to say it is coccus, or if there's plural, cocci. Instead of saying it's a rod shape, it's bacillus, or plural bacilli. Vibrio, yeah, there's actually bacteria that are comma shaped. H. pylori. Can't talk tonight. Not enough caffeine. H. pylori. Helicobacter pylori. Grows in your stomach. Causes ulcers. Shaped like a comma. And then, depending upon how tight the corkscrew is, a loose corkscrew, spirillum. A tight corkscrew, spirochete. Remember, this is still individual single cells that'll be elongated and have a corkscrew shape. Sometimes they're going to be long and thin. They refer to this as filamentous. You're going to find this with some of the soil microbes, soil bacteria. And then there's going to be bacteria that are all or none of the above. These are referred to as pleomorphic, pleomorphism. Now, irregardless of shape, bacteria can either be individual cells or they can appear in pairs called diplococci. They can also be as tetrads or come in clusters or chain or forming a cube. All of this is based on the division of the cell. Bacterial cells divide. One splits, becomes two. How they divide is determined by the accumulation of proteins which are under the direction of genes. So, one bacterial cell can divide, become two. If that's all that happens, this is a diplococci. When it's time for one of these bacteria to split, this diplococci, this pairing, will break apart. You'll have two individual cells that will continue to do so. Strep pneumonia, Streptococcus pneumoniae, is a known diplococci that's in your nose. Or if they stay together and they just keep making more and more longer chains, streptococci. Streptococcus aureus forms, Streptococcus pyogenes. It's Staphylococcus aureus. Streptococcus. Strepto means chain, coccus means sphere, a chain of spheres. Some bacteria have two planes. So not only will they split, one becomes two, but then those two will then split and you form this tetrad. If the cells genetically are predisposed to stay clumped together and then go through another division and you get 8, you now have a sarcina. And if this continues on and on and on, you can get a bigger and bigger cube. Remember, the plane of division is driven by genetics. Genes call for certain proteins to be expressed. Those proteins will then determine how the cell divides. And sometimes, as you see over here, from where I misspoke earlier, it's a random division, the random plane. You end up with this cluster. This is Staphylococci. Staphylo, Greek for cluster. The first microbiologist who looked at this under a microscope thought this reminded him of a cluster of grapes, hence the Staphylo, cluster of spheres.